Chapter 9 Biomolecules. There is a wide diversity in living organisms in our biosphere. Now, a question that arises in our mind is, are all living organisms made of the same chemical, that is, elements and compound? You have learned in chemistry how elemental analysis is performed. If we perform such an analysis on a plant tissue, animal tissue, or a microbial paste, we obtain a list of elements like carbon, hydrogen, and oxygen, and several others, and their respective content per unit mass of a living tissue. If the same analysis is performed on a piece of earth crust as an example of non-living matter, we obtain a similar list. What are the differences between the two lists? In absolute terms, no such differences could be made out. All the elements present in a sample of earth crust are also present in a sample of living tissue. However, a closer examination reveals that the relative abundance of carbon and hydrogen with respect to other elements is higher in any living organism than in the earth crust. Table 9.1. 9.1. How to analyze chemical composition? We can We can continue asking in the same way, what type of organic compound are found in living organisms? How does one go about finding the answer? To get an answer, one has to perform a chemical analysis. We can take any living tissue, a vegetable or a piece of liver, etc., and grind it in trichloroacetic acid using a mortar and a pestle. We obtain a thick slurry. If we were to strain this through a cheesecloth or cotton, we would obtain two fractions. One is called the filtrate, or more technically, the acid soluble pool, and the second, the retentate, or the acid insoluble fraction. Scientists have found thousands of organic compounds in the acid soluble pool. In higher classes, you will learn about how to analyze a living tissue sample and identify a particular organic compound. It will suffice to say here that one extract the compounds, then subject the extract to various separation techniques till one has separated a compound from all other compound. In other words, one isolate and purify a compound. Analytical technique, when applied to the compound, give us an idea of the molecular formula and the probable structure of the compound. All the carbon compound that we get from living tissue can be called biomolecules. However, living organisms have also got inorganic elements and compounds in them. How do we know this? A slightly different but destructive experiment has to be done. One weight a small amount of living tissue, say a leaf or liver, and this is called wet weight, and dry it. All the water evaporate. The remaining material gives dry weight. Now, if the tissue is fully burnt, all the carbon compounds are oxidized to gaseous form, CO2, water vapor, and are removed. What is remaining is called ash. This ash contains inorganic elements like calcium, magnesium, etc. Inorganic compound like sulfate, phosphate, etc. are also seen in the acid soluble fraction. Therefore, elemental analysis give elemental composition of living tissue in the form of hydrogen, oxygen, chlorine, carbon, etc., while analysis for compounds gives an idea of the kind of organic and inorganic constituent that is given in table 9.2 present in living tissue. From a chemistry point of view, one can identify functional group like aldehyde, ketone, and aromatic compound, etc. But from a biological point of view, we shall classify them into amino acids, nucleotide bases, and fatty acid, etc. Amino acids are organic compound containing an amino group and acidic group as substituents on the same carbon, that is the alpha carbon. Hence, they are called as alpha amino acid. They are substituted methanes. There are four substituent groups occupying the 4 valency position. These are hydrogen carboxyl group, amino group, and a variable designated as R group. Based on the nature of R group, there are many amino acids. However, those which occur in proteins are only of 20 types. The R group in this protein Proteinuses amino acid could be a hydrogen. The amino acid is called glycine, a methyl group alanine, hydroxymethylserine, etc. 3 of the 20 are shown in figure 9.1. The chemical and physical properties of amino acids are essentially of the amino carboxyl and the are functional group based on number of amino and carboxyl group. There are acidic, that is glutamic acid, basic lysine and neutral valine amino acid. Similarly, there are aromatic amino acid tyrosine, phenylalanine, tryptophan. A particular property of amino acid is the ionizable nature of ammonia and carboxylic group. Hence, in solution of different pH, the structure of amino acid changes. Lipids are generally water insoluble. They could be simply fatty acid. A fatty acid has a carboxyl group attached to an R group. The R group could be a methyl or ethyl or higher number of alkane group 1 carbon to 19 carbon. For example, palmitic acid has 16 carbon including carboxyl carbon. Arachidonic acid has 20 carbon atoms including the carboxyl carbon. Fatty acid could be saturated without double bond or unsaturated with one or more carbon-carbon double bond. Another simple lipid is glycerol, which is trihydroxypropane. Many lipids have both glycerol and fatty acids. Here the fatty acids are found esterified with glycerol. They can be then monoglycerides, diglycerides and triglycerides. These are also called fats and oils based on melting point. Oils have lower melting point, example gingerly oil, and hence remain as oil in winter. Can you identify a fat from the market? Some lipids have phosphorus and phosphorylated organic compound in them. These are phospholipids. They are found in cell membrane. Lecithin is one example. Some tissue, especially the neural tissue, have lipids with more complex structure. Living organisms have a number of carbon compounds in which heterocyclic rings can be found. Some of these are nitrogen bases: adenine, guanine, cytosine, uracil, and thymine. When found attached to a sugar, they are called nucleosides. If a phosphate group is also found esterified to the sugar, they are called nucleotides. Adenosine, guanosine, thymidine, uridine, and cytidine are nucleosides. Adenylic acid, thiamotylic acid, and guanylic acid, uridylic acid, and cytidylic acids are nucleotides. Nucleic acid like DNA and RNA consist of nucleotide only. DNA, RNA function as genetic material. 9.2 Primary and secondary metabolites. The most exist, exciting aspect of chemistry deals with isolating thousands of compounds, small and big, from living organisms, determine their structure, and if possible synthesizing them. If one were to make a list of biomolecules, such a list would have thousands of organic compounds including amino acids, sugar, etc. For reasons that are given in section 9.10, we can call these biomolecules as metabolites. In animal tissue, one notice the presence of all such categories of compounds shown in figure 9.1. These are called primary metabolites. However, when one analyzes plant, fungal, and microbial cell, one would see thousands of compounds other than these primary metabolites. Example: alkaloids, flavonoids, rubber, essential oil, antibiotics, colored pigment, scents, germs, spices. These are called secondary metabolites. Table 9.3. While primary metabolites have identifiable functions and play a known role in normal physiological process, we do not at the moment understand the role or function of all the secondary metabolites in host organisms. However, many of them are useful to human welfare. Example: rubber, drugs, spices, scents, and pigments. Some secondary metabolites have ecological importance. In the later chapter and years, you will learn more about this. 9.3 Biomacromolecule. There is one feature common to all those compounds found in the acid soluble pool. They have molecular weight ranging from 18 to around 800 daltons approximately. The acid insoluble fraction has only four types of organic compound, that is protein, nucleic acid, polysaccharides and lipids. These classes of compound, with the exception of lipids, have molecular weight in the range of 10,000 Dalton and above. For this very reason, biomolecules, that is chemical compound found in living organisms, are of two types. One, those which have molecular weight less than 1000 Dalton and are usually referred to as micromolecule or simple biomolecule, while those which are found in the acid insoluble fraction are called macromolecule or biomacromolecules. The molecule in the insoluble fraction, with the exception of lipids, are polymeric substances. Then why do lipids, for whose molecular weight do not exceed 800 Dalton, come under acid insoluble fraction, that is macromolecular fraction? Lipids are indeed small molecular weight compounds and are present not only as such but also arranged into structure like cell membrane and other membrane. When we grind a tissue, we are disputing the cell structure. Cell membrane and other membranes are broken into pieces and form vesicles which are not water soluble. Therefore, these membranes fragment in the form of vesicles get separated along with the acid insoluble pulp, and hence in the macromolecular fraction, lipids are not strictly macromolecule. The acid-soluble pole represent roughly the cytoplasmic composition. The macromolecules from cytoplasm and organelles becomes the acid-insoluble fraction. Together, they represent the entire chemical composition of living tissue or organism. In summary, if we represent the chemical composition of living tissue from abundance point of view and arrange them class-wise, we observe that water is the most abundant chemical in living organism. 9.4 proteins are polypeptide. They are linear chains of amino acid linked by peptide bond, as shown in figure. Each protein is a polymer of amino acid, as there are 20 types of amino acid. Example: alanine, cysteine, proline, tryptophan, lysine, etc. A protein is a heteropolymer and not a homopolymer. A homopolymer has only one type of monomer, repeating n number of times. This information about the amino acid content is important, as later in your nutrition lesson you will learn that certain amino acids are essential for our health and they have to be supplied through our diet. Hence, dietary protein are the source of essential amino acid. Therefore, amino acid can be essential or non-essential. The latter are those which our body can make, which while we get essential amino acids through our diet or food, proteins carry out many functions in living organism. Some transport nutrients across cell membranes, some fight infectious organisms, some are hormones, some are enzymes, etc. Collagen is the most abundant protein in animal world, and ribulose bisphosphate carboxylase oxygenase rubisco is the most abundant protein in the whole of the biosphere. 9.5 polysaccharides. The acid insoluble palate also has polysaccharide carbohydrate as another class of macromolecule. Polysaccharides are long chains of sugar. They are threads, literally a cotton thread containing a different monosaccharides as building block. For example, cellulose is a polymeric polysaccharide consisting of only one type of monosaccharide, that is glucose. Cellulose is a homopolymer. Starch is a variant of these but present as a storehouse of energy in plant tissue. Animals have another variant called glycogen. Inulin is a polymer of fructose. In a polysaccharide chain, say glycogen, the right end is called the reducing end and the left end is called the non-reducing end. It has a branches as shown in the form of cartoon figure 9.2. Starch forms helical secondary structure. In fact, starch can hold I2 molecule in the helical portion. The starch I2 is a blue in color. Cellulose does not contain complex helicals and hence cannot hold I2. Plant cell wall are made up of cellulose. Paper made from plant pulp and cotton fiber is cellulosic. There are more complex polysaccharide in nature. They have as building block amino sugar and chemically modified sugar. Example: glucosamine and acetyl galactosamine, etc. Exoskeleton of arthropods, for example, have a complex polysaccharide called chitin. These complex polysaccharide are mostly homopolymer. 9.6 nucleic acid. The other type of macromolecule that one would find in the acid insoluble fraction of any living tissue is the nucleic acid. These are polynucleotide together with the polysaccharides and polypeptide. These comprises the true macromolecular fraction of any living tissue or cell. For nucleic acid, the building block is a nucleotide. A nucleotide has three chemically distinct components. One is a heterocyclic compound, the second is monosaccharide, and the third a phosphoric acid or a phosphate. As you notice in the figure 9.1, the heterocyclic compound in nucleic acid are the nitrogen bases named adenine, guanine, uracil, cytosine, and thymine. Adenine, guanine are substituted purines, while the rest are substituted pyrimidine. The skeletal heterocyclic ring is called as purine, pyrimidine, respectively. The sugar found in the polynucleotides is either ribose, a monosaccharide, pentose, or 2-deoxyribose. A nucleic acid containing deoxyribose is called deoxyribonucleic acid. DNA, while that which contains ribose is called ribonucleic acid. 9.7 Structure of protein. Proteins, as mentioned earlier, are heteropolymer containing strings of amino acids. Structure of molecule means different things in different contexts. In organic chemistry, the structure invariably refers to the molecular formula. Example: NaCl, MgCl2, etc. Organic chemists always write a two-dimensional view of the molecule while representing the structure of the molecule. Example: benzene, naphthalene, etc. Physicists conjure up the three-dimensional views of molecular structure, while biologists describe the protein structure at four levels. The sequence of amino acid, that is the positional information in a protein, which is the first amino acid, which is the second, and so on. It is called the primary structure of a protein. A protein is imagined as a line, the left hand represented by the first amino acid and the right end is represented by the last amino acid. The first amino acid is called N-terminal amino acids. N-terminal amino acid. The last amino acid is called the C-terminal amino acid. A protein thread does not exist throughout as the extended right rod. The thread is folded in the form of helix, similar to a revolving staircase. Of course, only some portion of the protein thread are arranged in the form of a helix. In protein, only right-handed helix are observed. Other regions of protein thread are folded in other forms in what is called a secondary structure, that is given in figure 9.4b. In addition, the long protein chain is also folded upon itself like a hollow woolen ball, giving rise to the tertiary structure. These give us a three-dimensional view of a protein. Tertiary structure is absolutely necessary for the main biological activity of protein. Some protein are an assembly of more than one polypeptide or subunits. The manner in which these individual folded polypeptide or subunits are arranged with respect to each other. Example: linear strings of spheres arranged one upon each other in the form of a cube or plate, etc., is the architecture of a protein, otherwise called the quaternary structure of a protein. Adult human hemoglobin consists of four subunits. Two of these are identical to each other. Hence, two subunits of a alpha type and two subunits of beta type together constitute the human hemoglobin. HB 9.8 Nature of bond linking monomers in a polymer. In a polypeptide or a protein, amino acids are linked by a peptide bond, which is formed when the carboxyl group of one amino acid react with amino group of the next amino acid with the elimination of water moiety. The process is called dehydration. In a polysaccharide, the individual monosaccharides are linked by a glycosidic bond. This bond is also formed by a dehydration. This bond is formed between the two carbon atoms of two adjacent monosaccharides of one nucleotide to the fifth carbon of the sugar of the succeeding nucleotide. The bond between the phosphate and the hydroxyl group of the sugar is an ester bond. As there is one such ester bond on either side, it is called as phosphodiester bond, that is given in figure 9.5. Nucleic acid exhibit a wide variety of secondary structure. For example, one of the secondary structure exhibited by DNA is the famous Watson-Crick model. This model says that DNA exists as a double helix. The two strands of polynucleotide are anti-parallel, that is run in the opposite direction. The backbone is formed by the sugar chain. The nitrogen bases are projected more or less perpendicular to these backbone but face inside A and G of one strand compulsorily base pair with T and C respectively on the other strand. There are 2 hydrogen bonds between A and T, 3 hydrogen bonds between G and C. Each strand appears like a helical staircase. Each step of ascent is represented by a pair of bases. At each step of the ascent, the strand turns 36 degrees. One full turn of helical strand would involve 10 steps or 10 base pair. Attempt drawing a line diagram. The pitch would be 34 angstrom. The rise per base pair would be 3.4 angstrom. This form of DNA with the above mentioned silent feature is called bDNA. In higher classes, you will be told that there are more than a dozen forms of DNA named after English alphabet with unique structure. Structural feature 9.9. Dynamic state of body constitute concept of metabolism. What we have learned till now is that living organism, be it simple bacterial cell, a protozoa, a plant, or an animal, contain thousands of organic compound. These compound or biomolecules are present in certain concentration. One of the greatest discovery ever made was the observation that all these biochemical are turn over. This means that they are constantly being changed into some other biomolecules and also made from some other biomolecule. This breaking and making is through chemical reaction constantly occurring in a living organism. Together, all these chemical reaction are called metabolism. Each of the metabolic reaction result in the formation of biomolecule. A few examples of such metabolic transformation are removal of CO2 from amino acid, making an amino acid into an amine, removal of amino group in a nucleotide base, hydrolysis of glycosidic bond in a disaccharide ATC. We can list tens and thousands of such examples. Majority of these metabolic reactions do not occur in isolation but are always linked to some other reaction. In other words, metabolites are converted into each other in a series of linked reactions called metabolic pathway. These metabolic pathways are similar to the automobile traffic in a city. These pathways are either linear or circular. These pathways crisscross each other. There are traffic junctions. Flow of metabolites through metabolic pathway has a definite rate and direction like automobile traffic. This metabolite flow is called the dynamic state of the body constitute. What is the most important is that this interlinked metabolic traffic is very smooth and without a single reported mishap for for healthy condition. Another feature of these metabolic reaction is that every chemical reaction is a catalyzed reaction. There is no uncatalyzed metabolic conversion in living system. Even CO2 is dissolving in water. A physical process is catalyzed reaction in living system. The catalyst which hasten the rate of a given metabolic conversion are also proteins. These proteins with catalytic power are named enzymes. 9.10 Metabolic basis for living. Metabolic pathway can lead to a more complex structure from a simple structure. For example, acetic acid becomes cholesterol, or lead to the simpler structure from a complex structure. The former cases are called biosynthetic pathway or anabolic pathway. The latter constitute the degradation and hence are called catabolic pathway. Anabolic pathway, as expected, consume energy. Assembly of protein from amino acid require energy input. On the other hand, catabolic pathway lead to the release of energy. For example, when glucose is degraded to lactic acid in our skeletal muscles, energy is liberated. These metabolic pathway from glucose to lactic acid, which occurs in 10 metabolic steps, is called glycolysis. Living organisms have learned to trap this energy liberated during degradation and store it in the form of chemical bonds as and when needed. This bond energy is utilized for biosynthetic, osmotic, and mechanical work that we perform. The most important form of energy currency in living system is the bond energy in a chemical called adenosine triphosphate (ATP). How do living organisms derive their energy? What strategy have they involved? How do they store energy, and what is it and in what form? How do they convert this energy into work? You will study and understand all this under a subdiscipline called bioenergetics later in your higher classes. 9.11 The living state. At this level, you must understand that The tens and thousands of chemical compounds in a living organism, otherwise called metabolites or biomolecule, are present in concentration characteristics of each of them. For example, the blood concentration of glucose in the normal healthy individual is 4.2 millimole, while that of hormone would be nanograms per milliliter. The most important fact of biological system is that all living organisms exist in a steady state characterized by a concentration of each of these biomolecules. These biomolecules are in a metabolic flux. Any chemical or physical process moves spontaneously to equilibrium. The steady state is a non-equilibrium state. One should remember from physics that system at equilibrium cannot perform work. As living organisms work continuously, they cannot afford to reach equilibrium. Hence, the living state is non-equilibrium steady state to be able to perform work. Living process is a constant effort to prevent falling into equilibrium. This is achieved by energy input. Metabolites provides a mechanism for the production of energy. Hence, the living state and metabolism are synonyms. Without metabolism, there cannot be a living state. 9.12 Enzymes. Almost all enzymes are protein